Hello everyone, we are solving JAM 2024 paper of biotechnology, and here we are going to discuss around 5 to 6 questions, um, and also we are going to discuss all of these in quite detailed manner. So let's start with question number 6. Which one of the following is present in a bacterial cell? Now the thing that is present in a bacterial cell is, it would be 70S ribosome. Like, it means the presence of bacteria, presence in bacteria. 70S ribosomes, these are the hallmark of bacterial protein synthesis machinery. Okay, and bacteria lacking a nucleus and distinct organelles, they rely on these cytoplasmic structures for translation. Now, about the composition, these ribosomes comprise of two subunits. One is the smaller 30S, okay, 30S ribosome subunit, and a larger 50S subunit. So each subunit contains the ribosomal RNA, that's the rRNA, and ribosomal proteins. Now, specific RNA in bacteria. A bacterial 70S ribosome, they contain the 16S rRNA in the 30S subunit, and the 23S and the 55 and the 5S rRNA in the 50S subunit. So yes, importantly, they do not contain the 28S rRNA, which is found in the eukaryotic 80S ribosomes. Let's talk about the other options now. First option, incorrect option, is 28S rRNA. Now, this is not present in bacterial 70S ribosomes. It's found in eukaryotic 80S ribosomes. Okay. Hence, it's not the correct answer. Chitinous cell wall. Now, most bacteria have peptidoglycan cell walls, and chitin is found in fungi and some insects. Next is histones. Now, these DNA packaging proteins, they are found in eukaryotic nuclei, not bacterial cells. Okay. Now, talking a little bit about the ribosomal thing. Ribosomes are essential for all living organisms, enabling the protein synthesis from the mRNA templates, and the differences in ribosomal size and rRNA composition contribute to their distinct functions and evolutionary diversity. Studying these characteristics helps us to understand the protein translation mechanisms and to classify the organisms. Okay, next question. Which color of light excites a natural GFP to emit the green fluorescence? The very first one would be the correct answer, that is blue. The natural green fluorescent protein, that is the GFP, it's most efficiently excited by the blue light in the 390 to 480 nm wavelength range. When it absorbs the blue light, what happens? Its electrons transmit to a higher energy state, and upon returning to the ground state, GFP, it releases energy in the form of green light around 508 nm, resulting in the characteristic green fluorescence. Okay. Next, we are going to talk about the green color. Now, green light falls within the emission spectrum of GFP, meaning it will not excite the protein for fluorescence. Next is infrared. Now, infrared light has a longer wavelength and lower energy than the blue light, so it cannot excite the GFP effectively due to insufficient energy transfer. Next is red light. Now, red light has a longer wavelength and lower energy than the blue light, so it's similar to infrared. It's also outside the absorption spectrum of GFP and cannot induce the fluorescence. So yeah, we have discussed the correct answer. Moving over to the next question. Which one of the following hormones promotes the fruit ripening? Abscisic acid, auxin, ethylene, gibberellin. Your correct answer would be ethylene. Okay. While other hormones like abscisic acid, that is the ABA acid, auxin, and gibberellin, they play various roles in plant growth and development. Okay. But ethylene, that is the primary hormone responsible for promoting the fruit ripening. Okay. It is a gaseous hormone which is produced by fruits themselves only, and it triggers a cascade of changes that leads to softening, color change, increased sweetness, and other characteristics associated with ripeness. Point-wise, let's discuss abscisic acid or ABA. This plays a very complex role in ripening, sometimes promoting ethylene production but also influencing the stress responses and the seed development. Coming to auxin, auxin primarily it's involved in cell division, elongation, growth, and not specifically in ripening. Hence, it's not the correct answer. Coming to gibberellin, it can stimulate the fruit growth and development in some cases, but generally not associated with ripening. Hence, we have got our answer. Let's move over to the next question. Which one of the following has a catalytic RNA? Ribonuclease H, ribozyme, RNA polymerase 1, or RNA polymerase 2? Your correct answer would be ribozyme. That's B. Ribonuclease H. This is an enzyme, but not an RNA, if I say, and its function is to degrade the specific regions of RNA, not to act as a catalyst. Second, let's talk about our correct answer, that is ribosome. This is a specific type of RNA molecule that has catalytic activity, meaning it can accelerate the chemical reactions like protein synthesis. Next is RNA polymerase 1. Now, this is an enzyme that transcribes the rRNA, that is the ribosomal RNA, genes into the RNA. While the ribosomal RNA is involved in protein synthesis, it doesn't have catalytic activity itself. Hence, this will also not go. Coming to RNA polymerase 2. Now, this is an enzyme that transcribes the protein coding genes into the a messenger RNA, or what we call the mRNA, and similar to ribosomal RNA, these messenger RNA, they don't possess these catalytic activity. Okay. Therefore, only ribozymes have this catalytic RNA among the given options. Let's move over to the next question. The number of significant figures in a reported measurement of 361, it would be 3. Okay, go with 3. Let's start with the non-zero digits. They are always significant, right? And this includes the leading non-zero digit, that is 3, and the digit 6. Now, zeros between non-zero digits are again significant. Now, there are no zeros between 3 and 6, so this rule doesn't apply over here. Next would be trailing the zeros after a decimal point and a non-zero digit are significant. Now, in this case, what happens? There are no trailing zeros after 6. Therefore, the only significant digits are 3 and 6, resulting in a total of 2 significant figures. However, since the number is written in decimal form, we also need to consider the leading zeros. Leading zeros are generally not significant unless they are between a decimal point and a non-zero digit. In this case, the leading zeros are not between the decimal point and a non-zero digit, so they are not significant. Hence, the final answer is 3 significant figures, that is option A. Okay. Let's keep it till here in this video. If you need to know more about our classes and need to join, then you can Just check the description box below this video.